Hi, and welcome to the first installment of Life Science Life Hack, where I, Caroline Walker, teach all of you about the building blocks of life, also known as cells. In this episode, we are going to talk about the cell cycle. Alright, so the cell cycle is a very complex process, but we are going to break it down into four main phases. So, we're going to start with the G1 phase, or the growth 1 phase, and this, just like it says on the tin, is where the cell starts to grow in size. The next phase is the S phase, or the synthesis phase, where the DNA inside the cell's nucleus copies itself, so there are two identical copies of said DNA. Next phase is the G2 phase, where, just like in the G1 phase, the cell continues to grow and develop. The M phase, which is the shortest phase but arguably the most important, is where mitosis and cytokinesis occur, and the cell becomes two identical daughter cells in mitosis, or in the case of meiosis, four genetically unique cells. Next, in this process, we have a series of checkpoints and regulations to make sure that everything goes smoothly. There are three main checkpoints, or controls. The first one is the G1 checkpoint, also known as the restriction point. And this one is very important because it sends a signal that sends a go-ahead to start the entire cell cycle. G2, which is the next checkpoint, makes sure that duplicated DNA has been read over and repaired before mitosis occurs, so that way you can't have any weird mutations or anything that would trigger cell death or apoptosis. The next one is the M checkpoint, which makes sure that all of the chromosomes are lined up in the middle of the cell before anaphase can start, so that way each cell receives the appropriate amount of chromatids and DNA in order to function properly. This one specifically is an example of an internal signal, because all of the chromosomes have to be lined up internally inside the cell itself before this can occur. Now, we are going to talk about promoting factors, and specifically, we're going to talk about MPF and PDGF. MPF is known as the M phase, or maturation promoting factor. This one triggers mitosis itself, and this occurs after cyclins, which we are going to talk about a bit more later, accumulate to crucial points and bond to cyclin-dependent kinases, which we are also going to talk about more in detail. And this MPF turns itself off again during anaphase, which you can see in this picture down here. And the CDKs continue to bond again when the cycle goes back around, because this cycle is never ending. So the cyclins accumulate, bond, and then are destroyed. Accumulate, bond, and then are destroyed. Next, we have PDGF, as seen here, also known as platelet-derived growth factor. And this growth factor is very unique because in tissue cells, it allows the cell to completely skip the G1 checkpoint of the cycle, so it can just divide without any form of regulation. And is believed to help with healing wounds, which is why your blood clots very quickly to prevent you from bleeding out in any form. Alright, so now we are going to get into detail about cyclins and cyclin-dependent kinases, which I'm just going to call CDKs because it's much easier. So cyclins are proteins that regulate processes within the cell cycle. So they bind to cyclin-dependent kinases, which we will see in a moment, and they're very important because when they accumulate enough, they are the ones that give the go-ahead for mitosis and cytokinesis at the checkpoints of the cell cycle. So this is a CDK, and a cyclin will bond to that CDK and activate it. And when this occurs, these CDKs can activate and deactivate proteins that will control and regulate the cycle. And they are also very important because they're the ones that can give the go-ahead signals or give the stop signals. Now, if this gives a stop signal at the G1 checkpoint that we talked about earlier, for example, this will cause the cell to go into the G0 phase, which is where the cell just completely stops dividing in any form. And this can be found in the human body in muscle tissues. Now, we all hear about cancer, but it has some very fascinating properties, and one of these is that its cells don't stop dividing. In normal body cells, which have density-dependent inhibition, once there are enough cells, they will be triggered to stop dividing, so that way you don't have a bunch of unnecessary cells growing all the time. But cancer cells, they keep dividing. And so they are basically rendered immortal, which is why they are so dangerous, because they are almost impossible to stop. Now, these cancer cells undergo what is called a transformation because they can avoid the immune system's attempts to destroy them and start to form a tumor. So a tumor can either be benign or malignant. And when the tumor is benign, it stays right where it grew, and it doesn't really cause as many issues as malignant tumors, which have the ability to spread or move to any other tissues around the body. So they can grow and cover entire lengths of the body, which is very dangerous. So this has been the cell cycle and how issues in the cell cycle can cause cancerous cells. Hope that helps.